Hello students, welcome to EPG Pathshala. I am Dr. Varun Vyas from Department of Physics and Astrophysics, University of Delhi. Today we are going to talk on module Spectroscopic Instrumentation under the paper Measurement and Instrumentations. In this module, we will study about spectroscopy and its related instruments. Spectroscopy is the study of interaction of electromagnetic radiation with the matter. It is used for investigating molecular characteristics of samples of interest. In this module, we will study about two spectroscopic methods: UV-visible spectroscopy and infrared spectroscopy. First, we will study about the basic principle of measurement and the basic of instrumentation behind the technique. Spectroscopic methods are large group of analytical methods that are based on atomic and molecular spectroscopy. In general terms, spectroscopy is a branch of science that deals with the interaction of various types of radiation with matter. In this field of investigation started with the studies involved in interaction of electromagnetic radiation and the matter, but at present the field has been broadened to include interaction of matter and other form of energy. For example, acoustic waves and beams of particles such as ions and electrons. Therefore, the spectroscopy and spectroscopic methods refer to the measurement of intensity of radiation with the photoelectric transducer or other type of electronic devices. Most of the commercially available spectrophotometers are based on electromagnetic radiation. This type of energy can take several forms and is most easily recognizable as light or radiant heat. Based on the frequency and the wavelength range, the electromagnetic spectrum is classified into various classes. Gamma rays have smallest wavelength in the range of 1 picometer, and radio waves can have wavelength as long as 100 megameter. In this session, we will limit ourselves to UV visible and IR range. We will study about instrumentation and basics physics behind molecular absorption spectroscopy. Let us first study about ultraviolet and visible spectroscopy. Now we will look at the principle behind UV visible spectroscopy. UV visible spectroscopy falls into the category of molecular absorption spectroscopy. This technique primarily focuses on absorption characteristics of molecules within the electromagnetic spectral range of 160 to 780 nanometer. It is based on the measurement of transmittance or absorbance A of solutions contained in transparent cell having path length of B centimeter. The concentration of an absorbing analyte is linearly related to the absorbance. Experimentally observed transmittance and absorbance can be approximated with the following equations. T is equals to P solutions divided by P solvents. This is equals to P by P naught. This is equation 1. A, that is absorbance, is equals to log of P solvents divided by P solution. This is equals to log of P naught by P. This is equation 2, where P naught is the power of radiation passing through the solvent and P is the power of radiation passing through the analyte present in the solvent. The relation between the absorbance and transmittance is given by the following equation. A, that is absorbance, is equals to 2 minus log of percentage transmittance. This is equation 3. The extent to which absorbance will take place is mathematically explained by Beer's law, according to which A, that is absorbance, is equals to molar absorptivity E, B, that is path length, C, that is the concentration of the compound. This is equation 4, where measured absorbance is directly proportional to E molar absorptivity with units of L mole raised to power minus 1, centimeter raised to power minus 1. B is the path length in centimeter, that is width of the sample holder, and C is the concentration of the compound in the solvent, that is moles per liter. A limit to the Beer's law is that it can describe absorption behavior at relatively low concentration of analyte. At high concentration, usually greater than 0.01 molar, the average distance between the molecules responsible for absorption is very less. At high concentration, the molecules start affecting charge distribution of their adjacent molecules. This alters molecules' ability to absorb radiation at a given wavelength. As the extent of the absorption depends on the concentration, this phenomena causes deviation in the linear relationship between absorbance and concentration. Deviation in the Beer's law may also be due to molar absorptivity because it depends upon the refractive index of the medium. If any changes in the concentration can lead to alteration of refractive index of the solution, deviation in the Beer's law may be observed. Instrumentation. Instruments for measuring absorption in UV visible range of electromagnetic spectrum are made up of first, electromagnetic radiation source, second, wavelength selectors, third, sample containers, fourth, radiation transducers, and 5, that is signal processing and readout devices. For molecular absorption studies, a continuum source is required whose power doesn't change sharply over considerable range of wavelengths. Different types of sources are available in the market. They are hydrogen and deuterium lamps, tungsten filament lamps, and xenon arc lamps. Cells or cuvettes required for holding the sample and solvent are constructed out of special material that also allow radiation to pass through the spectral region of interest. They are normally made up of quartz or fused silica and are transparent to UV, that is below 350 nanometer, and visible region of the spectra. For visible region of the spectra, plastic containers can also be used. Two most commonly available spectrophotometers are single beam and double beam spectrophotometer. Single beam spectrophotometer consists of radiation source, filter or monochromator, match cells that can be interposed alternatively in the path of the radiation beam, a photodetector, an amplifier, and a readout device. The basic design of single beam spectrophotometer is shown in figure 1. These systems have interchangeable tungsten deuterium lamp sources. and are equipped with high resolution grating monochromator with variable slits. Photomultiplier tubes are used as transducers, and its output is digitized for printing, plotting, and storing data in useful form. The double beam spectrophotometer is similar to single beam, but there are additional beam splitters that can direct radiation to the reference and the sample cells simultaneously. This can be observed in figure 2. The signals from the two photodetector go to the difference amplifier and finally to readout device. They offer an advantage of compensating any short-term fluctuations from the radiation source and any drift in transducer and amplifier. They are also known to compensate for variations in the source intensity with the wavelength. Infrared spectroscopy. Let us first look into the principle behind infrared spectroscopy. The infrared region of the spectrum encompasses wavelength from 0.78 to 1000 micrometer. From the standpoint of application and the instrumentation, infrared is divided into near, mid, and far infrared radiation. Though the most commonly used range is in the infrared region is between 2.5 to 15 micrometer. A linear wave number scale, that is per centimeter, is preferred in infrared spectroscopy due to the directional proportionality between this quantity and both energy and frequency. In practice, it is the frequency of absorption of radiation that is responsible for molecular vibrational frequencies during the measurement. Absorption of IR radiation is largely confined to molecular species that have small energy difference between various vibrational states. If the radiation frequency exactly matches the natural vibrational frequency of the module, a net energy transfer takes place. This results in change in amplitude of the molecular vibration, and absorption of radiation is the consequence. Similarly, any rotation of asymmetric molecules around their center of mass can lead to periodic dipole fluctuation that interact with IR radiation. There is no net change in the dipole moment occurring during the vibrational or rotation of homonuclear species such as N2, Cl2, or O2. Compounds like these cannot absorb IR radiation, whereas all other molecular species can. Various kinds of molecular vibration can take place in a simple triatomic molecule, are shown in figure 3. Broadly, they are classified as stretching and bending vibrations. Treatment of vibrations. Molecular vibrations, like coupling of vibrations between atoms involving bonds to a single central atom, are analogous to mechanical model used in describing simple harmonic oscillator. The natural frequency of an oscillator can be described mathematically as mu nu m is equals to 1 by 2 pi into square root of k by mu. This is equation 5. where K is the force constant that depends on the stiffness of the spring. In our case, it depends on the strength of the bond. Mu is the reduced mass where two masses m1 and m2 are connected by a spring. That is, in our case, is a molecular bond, can be written as mu is equals to m1 into m2 divided by m1 plus m2. Let us assume that the transition in the vibrational energy levels are brought about by absorption of radiation, provided that the energy level of the radiation matches the difference in the energy level, that is delta E, between the vibrational quantum states plus the vibration causes fluctuation in the dipole. This difference in the energy level can be written as delta E is equals to h mu m. This is equals to h divided by 2 pi into root of k by mu, where h is the Planck's constant. The frequency of radiation nu can bring change identical to the classical vibrational frequency of the bond mu m can be derived from the equation 5 and 6 and is written as nu is equals to nu m. This is equals to 1 by 2 pi into root of k by mu. This is equation 7. The wave number mu bar is reciprocal of wavelength in centimeter and is directly proportional to the frequency and the energy of the radiation. Thus, it may be written as mu bar is equals to k mu, where k is the constant of the medium and wave number is reciprocal of velocity c of the electromagnetic radiation. Now, if we wish to express the equation 7 in wave number, then it can be written as nu bar is equals to 1 by 2 pi c into root of k by mu. This is equals to 5.3 into 10 raised to power minus 12 into root of k by mu, where nu bar is the wave number of an absorption peak in centimeter raised to power minus 1. K is the force constant of the bond in newtons per meter. C is the velocity of light in centimeter per second. And mu is the reduced mass. IR instruments. There are three types of infrared instruments available from the commercial sources. The first one is a dispersive grating spectrophotometer that are used primarily for qualitative measurements. Second is a multiplex instrument involving Fourier transform and suited for both qualitative and quantitative measurements. Third is non-dispersive photometer that have been developed for quantitative measurement of a variety of organic species in the atmosphere by adsorption, emission, and reflectance spectroscopy. Most widely used spectrophotometer for laboratory-based investigation is Fourier transform infrared spectrophotometers, also known as FTIR, because of their speed, reliability, and their convenience. The basic design of single FTIR spectrophotometer is given in figure 4. The majority of FTIR instruments are based on Michelson interferometer. An interferometer uses pattern found by waves to measure characteristics of the waves themselves or of the material that reflects, refracts, or transmits wave. Infrared sources, that is lamps, consist of inert solid that is heated electrically to a temperature between 1500 to 2200 Kelvin. A continuum radiation approximating to that of a black body is emitted from the source. Various kind of IR source that are used in IR instruments are Nernst glower. It is composed of rare earth oxides formed into cylinder of diameter 1 to 2 mm and length up to 20 mm. Next is a global source. It is a silicon carbide rod, usually of 50 mm in length and 5 mm in diameter. Next is incandescent wire source. It is tightly wound spiral of nichrome wire heated to about 1100 Kelvin by an electrical current. Next is mercury arc. It consists of quartz jacketed tube containing mercury vapor at pressure greater than 1 atmosphere. And next we have an ordinary tungsten filament lamp and a carbon dioxide laser source. Advantages: optics of FTIR instruments have much larger energy throughput, that is 1 or 2 order of magnitude, as compared to dispersive instruments. They have limited throughput due to use of narrow slit widths. It should be noted that interferometer is free of the problem of stray radiations because each IR frequency is effectively chopped at different frequencies. That means any radiation other than the frequency that is to be radiated onto the sample is removed by interferometer. FTIR is a very powerful instrument and that has extensive application in the field of chemistry required for analysis of unknown components in the sample of interest. This instrument is particularly useful for 1. Very high resolution work involving gaseous mixtures having complex spectra resulting from superposition of vibrational and rotational bands. Second, the study of analytes having high absorbances. Third, the study of substances with weak absorption bands, that is, compounds absorbed on catalyst surface. Fourth, for fast scanning investigations involving kinetic studies or detection of chromatographic effluents. Fifth, collecting IR data from very small samples. Sixth, obtaining reflection spectra. And finally, IR emission studies. In this module, we studied about two most important types of spectrophotometer methods. First, we studied about UV visible spectrophotometry, and we briefly studied about the Beer's law that describes the adsorption characteristics of molecules in the path of the UV visible radiation. Second, we studied about infrared spectrophotometry, where we studied about different types of molecular bending vibrations responsible for absorption of radiation at particular wave number or frequency. Thank you.